So, what do you remember from next lecture? Nothing. What do you remember from the next? No. I apologize. I'm waiting for news from a paper I just submitted. And it has been 2 weeks now, and it kills me. Anyway, what do you remember from last year's lecture? Last year. You can give up this course, okay? What do you remember from last week's lecture? What do you remember? Do you remember anything? I don't. Some kind of important concept. What is the clock? Where is the clock? How does it function? If I remember, I gave you a question of... the last thing we saw was that, it was observed that some gene, especially like this PER gene in Drosophila, had a rhythm of expression. It was going up and down every day. And then some scientist called Rosbach sort of say maybe if you remove PER, suddenly the expression of the mRNA sort of is blunted. So this is maybe because PER regulates its own expression, right? If you keep the messenger RNA but it codes for a non-functional protein, then the rhythm in the mRNA disappears. So this means that the functional PER protein is needed for rhythmic expression of the PER gene. This is the last thing we saw. But anyway, remind me what we have seen last week. That was only the last bit. What we have seen last week.
The hypothalamus plays a very important role in controlling the rhythms and also the suprachiasmatic nucleus.
Yes. The hypothalamus is important for controlling rhythm in the animal. Especially the hypothalamus is quite big. Especially two small group of neurons called the suprachiasmatic nuclei. They seem to be critical because when you remove them, you lose the rhythm. And then when you take suprachiasmatic nucleus from an animal and you transplant it into another animal, then the animal gets the characteristics of the transplanted SCN in terms of period length. So this is one of the last slides I showed you last time. This one, right? This one, right? So we saw this tau mutant hamster, we saw this wild type hamster, and if you take the tau is SCN and you transplant it into the wild type hamster, and then if you exchange the wild type hamster and you put it into the tau hamster, then the period completely changes. So if you have a normal hamster that gets the tau SCN, it becomes a tau hamster just because it has the tau SCN. So SCN is really critical for circadian rhythms. It seems to control, at least at the level of the behavior, the circadian rhythms of the animal. What else did we see? Some other concepts.
And it had to be multiple genes involved, because even if one was knocked out of the rhythm, it was still rhythmic, but it was...
That's right. There are probably other genes involved in the control of the clock. All that we know from the Mendelian ratio here is that it seems to be one single gene, but there is still rhythm there. Okay. That being said, it could simply be that actually this gene has not been completely destroyed. Maybe this gene is still functional, but the protein is a little bit different. So let's say if this gene code for an enzyme, like a kinase or something, it may be that the mutated kinase is more active than the normal one. And this will accelerate the clock. But if you completely break the kinase, maybe there is no reason. So it could be because there are more genes, or it could be because it's not completely broken. It depends. Okay.
Did they knock out the gene or did they just break it?
That's a very good question. This is not a knockout. It's a naturally occurring, well, laboratorily occurring mutation. And they noticed why these guys have a very strange period in hamsters. So they didn't redo mutations with genes. So they did not manipulate any gene. But we will see in a coming lecture that this is indeed a kinase. And still, what is the function of this mutation? What is the impact of this mutation on the function of that kinase is not very clear. This is actually what I am studying in the laboratory here. It's something related to this kinase, so we will see this one later. So yes, anyway, there are more than one gene possibly controlling the clock. What else? Yes, I was waiting for the last one there. Yes, so even though the SCN is important for the rhythms, oscillators, very nice word, oscillators were found as well in many other cells, like in the liver, in different tissues, even in cells in culture. Okay? So there seem to be clock in many different cells, not only the SCN. All right. And from this experiment, if you remember, this is we can see the two SCN, 1 and 2 here, and the expression of these mouse per 1 transcript. Transcript is another word for messenger RNA. The expression of this transcript was measured across 24 hours, many days, and then it was found that it oscillates. And this gene also, well, some genes like reverb alpha, whatever the name, DBP, HER2, were also found to oscillate in tissue culture cells. Okay, this is mammalian. Okay, this is mammalian. So there are many reasons. And in cells in culture, rhythms of gene expression of different genes were also found. And a long time ago, this was in Nature, I already showed you in 1990. Really one of the first time that oscillations of genes were found. And this was in Drosophila head. And he saw that this per gene oscillates in the heads many days in a row. And then when he breaks the protein, so still the mRNA is expressed, but the protein is non-functional, as I said. This completely kills the rhythm. But if you provide an exogenous functional protein, this rescues the original rhythm of the messenger RNA. So this experiment suggested that PER2 controlled its own expression. That the PER2 protein, that the PER protein is needed to impact upon its expression, the rhythm. But how? The question I asked you for today was, can you think about several cases in which PER could control its own expression? How could this happen?
Negative feedback mechanism?
Negative feedback mechanism, yes. Yes. Moreover?
Like with internal glucagons, so if one gets too high, then the production. It limits the production of the. Trying to think of the So when one gets too high, it limits the production of the other one, which stops that one being produced. So the levels go back down, meaning the one can increase.
Yeah, just a standard negative feedback loop. So negative feedback loop is, well, you have one A inhibits A. That's negative feedback. Okay? So maybe in this case we can draw. This is the representation of the PER gene in Drosophila, and this indicates the coding sequence. これからタンパク質の情報ね。And then from this gene, PER2 mRNA, why do I say PER2? This is in Drosophila. PER mRNA is produced, and then from this messenger RNA, the PER protein is produced. So the simplest negative feedback loop simply is that PER proteins inhibits directly its expression. But that's not the only possible. That's not the only possible situation. There are other situations. This is the one, as a scientist, you want to be correct, because it's very simple. But there are other cases in which this could happen. What else? Still with a feedback loop, it could also be positive. You don't know which is it. It could also be positive, that you need PER proteins to promote PER protein. But it's a little bit more difficult to envisage. So maybe it is easier to think, okay, if there is a lot of PER, they will start inhibiting and then it will go down. Then when the amount of PER protein is degraded, then there is no more inhibition, and then PER goes up again. And so it's easier to think that way. Positive, it's a little bit difficult, because if it was a positive feedback loop, then PER would just go like this. If nothing else is involved in the mechanism. The more PER you make, the more PER you make. Okay? So then oscillations will be more difficult to happen, more difficult to generate, if you only have PER. So, okay, let's just say it's only a negative feedback loop. But still, that may not be the only situation. This direct effect may not be the only situation. What could be the other situation?
PER can activate another protein or enzyme.
Yeah. It's an indirect feedback. So PER sort of activates another thing, another protein, or whatever, and then this sort of goes back on the PER. So it's plus or minus. So it's indirect. So PER controls some things that controls PER. What else? If you think not maybe only at the level of the molecules, but the level of the whole animal.
You receive the signal from the outer environment to change their phase.
Come again?
Maybe the PER will receive the signal from the outer environment, for example, light or food, and they will change their phase when necessary.
That's an interesting one. But so these flies who had destroyed PER2, the locomotor activity rhythm of these flies was also affected. So it's not only that the rhythm of PER2 was affected, but the locomotor activity of these flies was affected. And the fact that PER may react to the environment, it's difficult to understand, because if a fly is in a complete constant condition, still you can see PER cycling. Okay, in normal flies, you can see PER cycling. So there is no, if PER is still cycling in constant condition, it means that the input on the environment is not that important to keep PER cycling. And if you check the difference in the PER expression of these mutant flies who have no PER protein, or they have no functional PER protein, the rhythm is very strange in a light-dark cycle as well as in a constant dark environment. So it's difficult to join the environment into this, but you are very close. You are very close. It's something to do with somehow some also indirect mechanism, but even more indirect than this blue pathway. Even more indirect. Something that has to do not with the environment, but more with the activity of the animal. Because, as I said, the locomotor activity of flies with this mutant PER gene is also affected. Yes?
Anything to do with ATP breakdown?
No. It might be, we don't know, but no. So the last situation is, well, I can show you the slides. It's actually the last figure in this paper by Rosbach. So that's the first explanation. This screen is a little bit. Wow. So this is his last figure. So the first situation is that PER protein directly controls its own messenger RNA or its own gene. Okay? It could degrade its own messenger RNA, or it could promote or inhibit its expression. The second situation, which is the one you say, is that actually the PER proteins control something else that controls PER. And the last situation, which you don't see here, which is very annoying, but what you have here, actually, I will stop this guy. I don't know if you can see. This one. So, PER protein will sort of control something that will control the behavior of the animal. And the behavior, the rhythm of the behavior, will control PER expression. So that probably involves many genes, but it's an indirect effect. Something PER controls the behavior of the animal. The behavior is affected because PER protein is destroyed, so the influence of the behavior on PER is also affected. So it's a very indirect ways. Okay. Very indirect. So these are the three situations. And then that's maybe 10, 15, 20 years after this discovery. Maybe 10 years. People started to analyze the expression of all genes in different tissues. For example, here you have from a paper written by Hiroki Ueda, who is now in Tokyo. Now, there are many papers like this. What you see is an experiment in which the expression of rhythmic gene was studied. And I'm not sure if you are familiar with this kind of heat map picture, but what you have at the bottom here, it's the time of the day. 0, 12, 24, 36, 44. This is in a light-dark cycle, and this is in constant darkness. This panel is for the SCN, the suprachiasmatic nucleus, and this panel is for the liver, and each line is one specific messenger RNA, one specific gene. So it means that many rhythmic genes were found. Actually, as the title says, if you look at the whole genes expressed in the liver, about 10% are rhythmic. So here you have the 10% that are rhythmic. And you cannot see the numbers of the gene expression, so it's a semi-quantitative way. Well, actually it's quantitative, but the amount of RNA is represented as a color. So a lot of messenger RNA means red, not a lot of messenger RNA means green, and average level means black. So you can see, so you have two cycles, like 0 to 24, 24 to 44. And you can see these genes are very high during the day, but then there is a change of phase. These genes are very high during the night, and these genes are high a little bit later at night. In the SCN, in the liver, many more genes were found to be rhythmic. You can see there is many more lines than this. But you can see here, there are genes that are expressed very early during the day, and then genes that are expressed later during the day, genes that are expressed earlier during the night. So you have many different phases of gene expression. Very different phases. Very different phases. And here you have the distribution of the circadian phase. This is circadian time. So this is the peak to 0 to 4, this is a peak to 4 to 8, peak to 8 to 12, and this is less important. But you can see very clearly that there are many different phases. There are genes that are high during the day, there are genes that are high during the night. And now what I can tell you is that it is true that PER regulated sound expression. That is true. And that also has been found in mammals. There are more PER than in Drosophila. In mammals, there are 3 PER proteins. And they control directly their own expression. And as you can see from the slide before, PER has a very nice phase. Very nice phase of expression. In the flyhead, it's high during the night. In the SCN, some genes are indeed high during the night. But, okay. It is easy now to understand how PER can control its expression. But here, we have many genes that are rhythmic. How could this be generated? Can you imagine a way by which genes will oscillate with the peak during the day, genes will oscillate with the peak during the night? Can we make a clock out of your ideas? So the basic feedback loop has been established. The basic feedback loop has been established. Now we are going to complete this schema and end up at what is known today. So, how can we go from this very single feedback loop, where per controls its own expression, to this? So that's essentially just the clock, negative feedback loop. That is molecularly what is the biology data. It's a negative feedback. But somehow, this negative feedback loop leads to that. So how can we make a clock with what is here to actually account for this observation?
For example, when you're with the highest value, then there will be some kind of hormone secreted into the blood, and it will control all the activities.
Yes. When PER2 is high, there might be some hormones secreted in the body. Yes, but how? Maybe it's true. Maybe some of these genes here actually code for a peptidic hormone. And then one of these hormones, that is maybe somewhere here, maybe insulin or whatever, will be under the control of per gene and sort of control of the genes. But many different phases. Many different phases. Not only genes during the morning, but also genes at night, and different times during the night as well. And then how per may control hormone expression. How could we devise a way? It's true, you are right. Per also controls other things than itself. This you are right. But not necessarily hormone.
Is it only like a negative feedback loop, so that that activates another one which activates like a cascade?
Like a cascade of negative feedback loop.
Yeah.
So, what do you mean? So, if we stay there, so we have per proteins, and then what happens?
And then per either activates or inhibits another protein, and then that protein, a gene, and then that activates another one.
Yeah, okay, that's what's happening. That's exactly what's happening. So not only, this is also what you said, specifically with the hormone, then you imagine all the genes. I will write N to mean it's many genes as we can see. They are also themselves inhibited by PER2. What is the consequence of that in terms of expression of these genes? If these genes are regulated in the same way than PER, what is the consequence on the expression of these genes?
They will, like, their pattern will be exactly the same as per gene.
What pattern?
Like when they peak during the day, like the rhythm.
How is it called?
How what?
How is it called? There is a specific term in oscillations about that. It's not exactly the wave, well, it's the waveform, but there is a specific, how would you say? How would you say in English? You have many words for the oscillations. You have the period, this is 24 hours. You have the amplitude, this is like, maximum divided by minimum divided by 2 or whatever. And then what else? You are right. What else? What words? The phase. The phase of expression. So if PER has this phase, so need of black marker. If the pearl has this phase, let's say in DSCN, okay? So this is the day, and this is the night. If pearl has this pattern, and it is regulated by itself, then all these genes that are also regulated by pearl should have roughly the same phase. The same phase of expression. We are making progress. That accounts for about a fourth of the genes we see here. There are many genes who are expressed with a completely different phase. Maybe we can see this here. Maybe these genes here might be controlled directly by pearl. What about the genes that are expressed during the day?
Like pearl controls different genes, but like different concentrations of pearl has different effects on different genes.
Yes, that's a good point. That's a good point. But that might affect something like the amplitude of oscillations. The amplitude of oscillations. Your idea was interesting. You say a kind of cascade of feedback.
So just the genes that activates that control the others.
Exactly. So this is where we have to think of other clock genes than pearl. So not only pearl will regulate some genes, but among these genes there will be other clock genes that will regulate, like can be positive or negative, other genes. So it means that the clock is not only one feedback loop, it's interlocked transcription, translation, feedback loop. So let's see in more details what I mean. And this is a very small picture that shows really the essential of the circadian clock. Slightly it becomes a bit complicated. So this circadian transcriptome, do you know the word transcriptome? Have you come across it before? Transcriptome means the entire messenger RNAs within the cell. The whole thing. And when you say circadian transcriptome, it means all the messenger RNA that have a rhythm. The whole messenger RNA that oscillate. Maybe as I say, 10% of the transcriptome is circadian. So, this is in mammals. Okay? This is Hastings, a guy from Cambridge, really nice guy. He likes football a bit too much, but we already know this pearl. And it's in mammals, and it's in 2007, so not so old, but still it has made a lot of progress. So we have pearl, and it's pearls in mammals, because as I said, in mammals they are pearl 1, pearl 2, pearl 3. Pearl regulates its own expression. This is the negative feedback loop of pearl. But it's an indirect control. It's direct, but it's indirect. So what happens is pearl, as well as other genes called cry, it's a different word, cryptochrome 1 and cryptochrome 2, they have the same phase, roughly. And what happens is that these genes, cry and pearl, their expression is activated by two other proteins called clock and BMAL. This becomes complicated. So clock and BMAL, do you know what it means? Transcription factor. Do you understand the word transcription factor? No? Then I need to explain this, because otherwise the rest of the lecture will go down the drain. So forget this for the moment. I think it was a bit too much of a jump. Okay. Let's go back to this single negative feedback loop. When I say that per regulates its expression at the molecular level, how can this happen? How can a protein regulate its own expression? The most direct way we can imagine.
The protein part binds to the promoter region of the gene, and then it activates the transcription.
Okay. Yeah, exactly. Everybody understand what is the promoter region of a gene? A gene promoter? No. Okay. So, a gene is essentially composed of two parts. Well, there are more than two parts, but the part that is indicated in textbook like this is the beginning of the transcription. From this to the end of the gene, let's say this, the mRNA will be produced. This is the mRNA with the poly A tail. And a small part of the messenger RNA will be used to make the protein. Okay? So this part is called the coding part. This is the part of the gene that codes for the messenger RNA and the protein. But it's more precise than that, but I try to be as simple as possible. But this part here is also very important. Because although it is not made into messenger RNA, it is like a control box for the expression of this gene. In the DNA here, there is a sequence. This is DNA, right? So it has a specific sequence. And that sequence will be very important to control the expression of the gene. Some sequence, maybe, I don't know, ATTA, will be very important to activate the gene. Some other sequence, like GACC, it's just completely random, okay? May be very important to inhibit. Okay? This is called the promoter region. Promoter region. So the promoter region contains specific sequence of DNA that have a regulatory function. They will control whether the gene is expressed or not. That's great, but DNA is only sequence. DNA by itself doesn't do much. It's just coding for information. So this control happens because imagine this is the per promoter. So we have the per protein here. We have the per messenger RNA. And in the promoter of per, let's say per 1 in mammals, there is a specific sequence that is CAC. This is not at random, this is the real sequence. GTG. Actually, there are 5 of these sequences, every 500 base pairs. CACGTG. And that it was found that this sequence is important to promote per expression. This sequence is important to promote per expression. But as I said, this is only a sequence. it doesn't do anything by itself. It's just a sequence of DNA. Actually, this sequence, it's called the E-box. E-box. E means enhancer. It's a box that enhances the expression of PER. It's the enhancer box. And later it was found that this E-box is bound by two proteins. Some proteins in the cell, they go into the nucleus and they recognize the CSC-GTG and they bind to it. So there are two proteins who have a structure that is great to bind to this CSC-GTG. And these two proteins are CLOCK and BMAL1, which are written there. Here, CLOCK and BMAL1. So, when CLOCK and BMAL1 bind to the DNA, they activate the expression of PER gene. This is what we see here. CLOCK and BMAL activate PER gene. Not only PER, but also CRY genes and many other genes. All genes that have E-box in their promoter are actually activated by CLOCK BMAL because they have the same sequence. Okay. So then what happens is that when CLOCK BMAL bind to the PER promoter, PER transcription is activated, protein is produced, and then this is where the feedback happens. PER inhibits the activity of CLOCK and BMAL. PER is an inhibitor of CLOCK and BMAL. So what happens is that CLOCK and BMAL activate PER and CRY, but let's focus on PER. And then when the level of PER protein increases, they inhibit the activity of CLOCK and BMAL. So everything is here. CLOCK and BMAL activate PER, CRY, and then they feedback. Okay, why there is a CRY here? It is because it was found that PER does not act by itself. It needs to associate with another protein called CRY. So this is PER. It becomes a little bit complicated. This is PER, and I write PERs because it's many of them, and they associate with CRYs, and together they go into the nucleus and they inhibit CLOCK BMAL. So this is what you have in this blue part here. CLOCK and BMAL activate CRY and PER. They form a complex, and together they inhibit their own expression. Okay. So that's only one part of it. Is everything okay so far? It becomes a bit complicated. Okay? All right. So, that's only one of the loops. That's one loop. But other loops were found. Almost at that time, you can see 2007. So between 2000 and 2007, almost one loop was discovered every year or something. So what's also happened is that CLOCK BMAL also activates the expression of other genes, reverb alpha and ROR. These, if they are activated by CLOCK BMAL, what does it mean? If I tell you that CLOCK and BMAL activate the expression of reverb alpha and RORA, what does it mean? From what I explained before, what is the consequence of that? What conclusion can you draw? clock and BMAL activates the expression, of reverb alpha and RORA.
They're in the same phase.
They're in the same phase, yes. They have the same phase as per and cry. But what does it mean? Why they have the same phase? Because they're activated by clock and BMAL. But why? In terms of the sequence, in terms of what do they have in their promoter? If reverb alpha and RORA are also activated by clock BMAL, what conclusions can you draw about their promoter?
It's the same.
Yeah. Reverb alpha and ROR alpha, they are also E-box control genes. In their promoter, they also have an E-box. But they are different from per and cry. Here, even though they are controlled by clock and BMAL, they will not inhibit their own expression, or they will not inhibit the expression of per and cry. They will actually regulate the expression of BMAL1. Reverb alpha acts like an inhibitor. Reverb alpha will be activated by clock and BMAL and sort of inhibits the expression of BMAL1. ROR alpha, or RORA, whatever you want to name, actually will not only activate BMAL1, but will also inhibit yet another couple of genes. This becomes very difficult. This is DBP and E4BP4. So this is the ancillary loop. Ancillary means additional loop. So DBP is also E-box control gene. You can see it is controlled by clock and BMAL. So they are controlled by E-boxes, but they are also controlled by reverb alpha and RORA. So in the end, all the loops are interlocked and they regulate each other. So especially you can see reverb alpha and RORA, they both regulate BMAL1. So it means that the relative amount of RORA and reverb alpha will affect BMAL1. And only at a specific time, when RORA is low and reverb alpha is high, BMAL will be inhibited. And when RORA is high and reverb alpha is low, BMAL1 will be produced. So all these are interlocked into each other. And this whole thing, all together, they will regulate essentially 3 different groups of genes. They are genes that are controlled. So these are the output genes. Not the genes of the clock, but other genes, like these guys. Like these guys. Some of these genes will be genes that are sensitive to the E4BP4 DBP couple. Some genes will be genes that are regulated by the E-box. And some genes will be genes that are regulated by the ROR. And why they are regulated? Why some genes are regulated by E-box? Why some genes are regulated by D-box? And why some genes are regulated by RORE? It is because each of these are transcription factors. They regulate DBP, E4BP4, they are binding to DNA, but at different sequence, not at CAC GTG. DBP and E4BP4, they bind at DNA. It's a sequence called D-box. It's a very long sequence that is in some promoter. So when this D-box sequence is present in the promoter, then DBP and E4BP4 will regulate these genes. Genes with E-box, we know already. This is CAC GTG. And RORE, this is ROR element. These genes contain a specific sequence that is bound by reverb alpha and RORA. And because of that, they will all have a different phase. Each of these clock genes is expressed with a different phase. So the phase of each clock gene, it's the synthesis of all the elements, D-box, E-box, RORE, that exist in their promoter. So for example, if you have a gene that is only regulated by a D-box, for example, it becomes very complicated. So I told you that cry and purr more or less have the same phase. Actually, it's not exactly true, because maybe you have whatever the phase, maybe the purr is like this, but cry is like this, a little bit later. This is because in the promoter of purr, not only there is an E-box, but there is as well DBP. And it comes a little bit earlier. In the cry gene, there is only an E-box. So then it is a little bit later. But this becomes a bit complicated. But still, we don't completely understand how it works. We don't completely understand what is all the interaction between these guys. But this is what happens. The phase of expression of these genes, whatever the phase, is actually controlled by all these genes together. Okay. And now when we have these many loops that interlock into each other, then we can have temporal organization of the whole transcriptome like this. And then we can even go further. What these guys have done is that here, it's now easier to understand. So this is the one I showed you before, with some examples shown here, whatever. But then what they have done is that they have seen, okay, these genes here, they have the same phase as ID2. ID2 is yet another clock gene, okay? Doesn't matter so much. But let's just focus on this one. These genes, they are like BMAL1 phase. These genes have the same phase of expression as BMAL1. These genes have the same phase of expression as CRY1. And these genes have the same phase of expression as reverb alpha and PER2. So what does it mean? These genes have the same phase as BMAL1. By what will they be controlled mainly? By what clock genes these genes with the same phase as BMAL1 will be controlled? From the previous slides, I showed you. Not the E-box. Not the E-box. The E-box will be this one, like PER2. The genes that have the same phase as PER2 will be controlled by the E-box. Reverb alpha as well has an E-box. But the genes that have the same phase as BMAL1, actually BMAL1 is completely, this is PER, and this is BMAL1. It's completely in antiphase, as you can see here. So by which clock genes will these BMAL1-like phase genes will be controlled? Yes. The BMAL1-like phase will be controlled by the reverb alpha R1 couple. The CRY1 will be maybe in between, I don't know, only E-box and some other things. So there are different phases according to which elements they have in their promoter. And in the SEN and the liver is a little bit different. In the liver we have reverb alpha phase, we have ID2 phase, we have a PER2 phase, and we have a BMAL1 phase, right? Yeah, we have a BMAL1 phase, and we have a CRY1 phase. So it's a little bit different. And hang on a minute, there is a difference. Okay, the BMAL1 phase here is genes that are high MAP during the night. And genes that are in the liver, they're also high during the night. Right? they are BMAL1 phase, they are high during the night. So that's okay. Genes that are same phase as reverb alpha and PER2, they are high during the day here. And the genes that have PER2, they are high during the night. Okay, it's a bit different. Okay. So if you take PER2-like phase in the SCN, these genes, there you can use this part here, right? This is the same scale. So this first bit here from 0 to 12 is the day, and 12 to 24 is the night. The genes that are like PER2 in the SCN, what they tell us here, is that these genes are high towards the end of the day. But in the liver, the genes that are like PER2, and therefore PER2 itself, is much later. It's during the night. So we don't understand everything. What I told you, yeah, this is the molecular oscillator, but it's different.
It takes time for the signal to trace these patterns from the SCN to the liver.
Exactly. This is how we understand it, is that there is a delay between the SCN and the peripheral clocks. Actually, the SCN controls the clock in other tissue. But it does not explain everything. Because even though you take other tissues, you take them out of the body, the phases between the genes will be different between the SCN and the liver. That's true, there is a delay. So the SCN always sends signal via hormone and via nerve signal to different tissues to say, okay, you have to keep on ticking. But the phase relationship between the genes is very difficult to understand. We still don't really understand why. We still don't understand why. And as a rule, and you notice perfectly, is that the phase of the genes in the periphery, so liver, skin, muscle, will be delayed compared to that of the SCN. But we don't completely understand why. We don't completely understand why. It has to do with communications between the tissue, as you correctly mentioned, but not all of it. Because, again, if you take them out of the body, and then still you will have difference in the phase between different genes. So one thing is that actually, if you quantify the expression of PER2, PER1, clock, BMAL1, all these genes, they have different levels in different tissues. They are not always expressed at the same level. So what I mean by level is that for example, let's say PER1 and PER2. Let's compare PER1 and PER2. If you go into the SCN, PER2 has a very nice high amplitude rhythm. And maybe the baseline of the gene, let's say, it's just a comparative figure, maybe 10. But if you check in the liver for PER1, then PER1 suddenly has very low baseline, 1, so it means the expression is much lower, and the oscillation is very weak. So each tissue is different, because they express different clock genes at different patterns. And this is because there are different transcription factors in tissues. You know, the liver is the liver because it is the liver. It has many different genes expressed compared to the SCN. So because of that, the clock is a little bit different as well. PER2 is the opposite. PER2 in the liver is very high amplitude but in the SCM, not so high. So there are different things like this. We still don't completely understand why this happens. Actually, I have one of my students, he's currently investigating this a little bit. And we have an idea, is that I will come later with this, but there are two other clock genes which I will mention later, and what we discovered here, not so long ago, is that, no, it's a little bit too complicated to say that now. Essentially, yeah, you still don't understand how it works. This is what it means. Alright? So, this is, now you're very familiar with this. This is work done by Okamura Hitoshi here, many years ago. This is locomotor activity behavior of a mouse. Normal mouse, wild type mouse. And this is the locomotor activity of a knockout mouse who lacks cry1 and cry2. So it lacks two clock genes, and then these mice are completely arrhythmic. In constant darkness, they have no rhythm whatsoever. So, if we go back to the simplistic model there, so if you remove all the cry together, you lose the clock. The clock completely disappears. If you also destroy per 1, per 2, per 3, the clock also disappears. If you only lose one of the cry 1, or only lose cry 2, or only lose per 1, or only lose per 2, still you have rhythm. But the period will be different. Just like the toe hamster that we saw. To lose the rhythm, to completely lose the rhythm, not simply having a different period, then you must remove all the homologues of cry, all the homologue of purse. The only gene who has a homologue, but deleting only one of them is enough to break the clock, is BMAL1. This is the only one. BMAL1, there is also BMAL2. But BMAL2, we don't know its function at all. It doesn't seem to be linked with the clock at all. If you destroy BMAL1, you completely lose the clock. Completely lose the clock. All right. But gene expression and protein synthesis is usually very fast. Can occur within minutes. Some genes, they can be expressed within 10 seconds, within 30 seconds. Gene expression and protein synthesis is usually very fast, can occur within minutes. So one cycle of the clock may only take 2 hours. In terms of biochemical reaction, 24 hours is very long. What is missing? If you only think that the clock is gene expression, so clock genes regulating the expression, how can it last 24 hours? Translation is very fast. The enzyme that makes protein, well, ribosomes and all these ribosomal proteins and ribosomal RNA, they work extremely fast. Transcription works extremely fast. But one cycle of the clock is 24 hours. Theoretically, this loop per clock and BMAL activating, per being produced, inhibiting, this can happen in minutes. But it lasts 24 hours. If you take per gene, and you don't count the oscillations, the time between peak and peak is 24 hours. So something is missing. Again, something is missing. There are more levels than just simply transcription and translation. What could there be? What could there be to make the clock? Actually, there is a clock that is 2 hours of period. When we are barely past the egg, when we are still, we look like a shrimp. The embryo development. Drosophila is the same. Actually, at that time, in a specific part of our body, which are called the somite, have you seen the somite? So these are the segments in a very early embryo. There is a clock that ticks and has a period of 2 hours. Very, very quick, fast oscillations. Similar function than the clock here, but 2 hours oscillation, so very fast. Here we have 24-hour oscillations. It's extremely long for something that is mediated by transcription or translation. Too long.
We have to consider the activity of the interaction between the genes expression and the activity of humans.
So you mean between the expression and the activity of what? Of the protein?
No, for example, when the genes reach a critical value, then a hormone is released into the blood, and it will control the activity of the human, and it will take a much longer time.
It's an interesting idea. But even if you take a cell, there is no hormone. You look at a single cell, still the oscillations of that single cell will be 24 hours. So what is missing? Well, there are many situations, there are many possibilities. There is not only one answer here. There are many things. There are things that we know and things that we still don't know. What could be? So what seems to be happening is that what you said is correct. There seems to be a delay. There are mechanisms that delay things, or that make sure that things happen at the right time. So in this reaction, this biochemical reaction, they are very fast, so they need to be delayed. How can they be delayed to tick with a 24-hour period? That is the big question. And there are many ways. There are many ways to achieve that biochemically.
Proteins bind DNA so weak, so many protein needed.
Yes, that's interesting. That's a possibility that maybe the binding, say, of BMAL1 to the E-box is not very strong, so you need to have a lot of BMAL1 to finally activate. Okay, but then, yeah. So that's one possibility, that the affinity is regulated. That's very true, and that is especially true for an organism that is not mammals, which I will see later. There is an organism. Do you know cyanobacteria? Cyanobacteria, they are just like photosynthetic bacteria, and their clock is not transcription translation feedback loop. Their clock is phosphorylation cycle. It's 3 proteins that phosphorylate each other. And the main guy in this research is a guy from Nagoya. And he has discovered that actually as a kinase, they are not very efficient. They have a very slow reaction rate to make sure that this happens in 24 hours. And they have a specific domain of the big protein, I will see that later, to actually delay the reaction rate. So this is a very good idea. What else? And the affinity of all these proteins is controlled. Yeah, that's one thing. What else?
Maybe there's some access and competitive inhibitor enzyme, and so it takes longer.
Yeah, that's a little bit similar. Is that things that can bind to proteins and that can protect and prevent the binding with other proteins. That's another possibility. What else? So the affinity. What else?
Time the protein can survive in the cell.
Exactly. More specifically, the stability of the proteins. If the proteins are very unstable, they will be very quickly degraded. And you can imagine that oscillations of PER, for example, which maybe take 12 hours, if they are very unstable, then within 1 hour they will be completely destroyed and the clock will be much quicker. So the stability of all these clock proteins is very tightly regulated. Very tightly regulated. But not only the stability of protein, what else?
The mRNA?
Yeah, the stability of the messenger RNA also is regulated, absolutely. So, this is the last thing I'm going to see today. Of course, let's make the thing more complex. Let's play with kinases and ubiquitination. Do you know what is ubiquitination? What is a ubiquitin? 聞いたことある? Ubiquitin.
That binds protein.
Yeah, what is ubiquitin? What kind of molecule it is ubiquitin? What kind of molecule it is ubiquitin?
Protein?
Yeah, exactly. It's a peptide, it's a polypeptide. It's a polypeptide that is added to a protein. And what is the role of this peptide, of this ubiquitin?
It says it is a garbage.
Yes. Please degrade this. This is what ubiquitin is. Ubiquitin is like a label. You know, when you have a big garbage in your house, you have to go to the company and ask a label. This is to take away. And then you put in front of your house with the label. This is the ubiquitin. Please take it away. Please degrade. So, ubiquitin is one thing. So we have, now you know all these things. So you have reverb and ROR, which control RORE element B ring gene, like BMAL1. BMAL and clock, they activate the expression of ROR, reverb alpha, PER1, PER2, cry1, cry2. And then PER2, ROR alpha and reverb alpha, they are translated into protein. They feed back and control the expression of BMAL1, which then affects their own expression. And then the PER proteins and cry proteins, they also form complexes. and they feed back into the nucleus to inhibit their own expression and expression of other EBOX media, control genes. But that's not all. As you can see, PER and CRY, they can be degraded. PER are degraded because they are phosphorylated. Phosphorylation, Lin-San, they can be phosphorylated by another gene that is CK1 epsilon or delta. This is casein kinase 1 epsilon, casein kinase 1 delta. And when they are phosphorylated, they can be degraded very quickly. And then CRY is the same. CRY can be phosphorylated. We don't really know much by what, but it can be ubiquitinated by FBXL. So this is a protein that adds ubiquitin to CRY, and then they are very quickly degraded. But also, somehow we still don't understand what happens. Phosphorylation at other amino acid of CRY and PER has a different function, which is to promote the entrance of CRY and PER into the nucleus, and then inhibits EBOX. And in the nucleus, of course, CRY and are also degraded. So many different levels. Very, very, very complex. Extremely complex. And this was maybe like a few years ago, and a lot of experiments and a lot of results on what happens when these proteins get phosphorylated and the stability of this protein. But what is a big focus nowadays is what happens before. What happens at the RNA level? How are all the RNA of these ROR alpha, reverb alpha, PER, how is it regulated? This is a currently very hot subject in chronobiology. Something interesting is that these guys, the tau mutant hamster, is actually a mutation of casein kinase 1 epsilon. This kinase is mutated, and what we think is that this kinase is more active. So it degrades very quickly PER, they are quickly degraded, and the clocks become shorter. This is currently how we think about it. And the very first human mutation to be discovered, I don't know if you have heard about FASPs. Red. This is nice. FASPs. Have you come across FASPs? Some people have FASPs. FASPs is familial advanced sleep phase syndrome. Familial advanced sleep phase syndrome. So these people have a short period, just like the tau mutant hamster. Every day they want to sleep at 5-6 o'clock in the afternoon. And at 3-4 o'clock in the morning they want to get up. Their clock runs very fast, and they have a lot of trouble to adjust to the normal light-dark schedule that we have no problem with. And these humans, these patients, two mutations were discovered that cause FASPs. One was a mutation in the PER gene, PER2, and one was a mutation in CK1 delta. And the mutation in PER was a target of CK1 delta. So both these human mutations, they were both something to do with casein kinase 1. So casein kinase is extremely, extremely important for the clock. It's really one of the key things. Unfortunately, if you remove casein kinase 1 delta from the animal dies. That annoys me because this is my research, the CK1. So this is very important kinase that regulates many things. Not only the clock, but many other things. It's involved in. It has been involved in cancer development. It has been involved in cell cycles, so really, really many fundamental things. And the only human mutations known is really this CK1D target site on PER or CK1D mutation. So that's the last thing I see today. Thank you very much. And this figure is actually made by, you don't see it here because it's like cut, but it's a guy called Joe Takahashi. He is American. Nice guy, one of the big guy in circuit immunology. He's close to retirement now, but yeah, Joe Takahashi. Very big name. That's it. Many things today. Many things. You saw the slides. Did I put the slides on Culasys? Did you download the slides from Culasys?
I can't access it yet because I'm Exchange.
This is really bad. I'm going to say something to the organizers. You cannot download. Who has downloaded the slides from Culasys?
No.
Hello? I think I uploaded them, I think. Maybe I forgot, which is very, very likely. Let me confirm that.
No resources.
No resources! Oh my god, I'm terrible. Your classes? Where am I? Introduction to Biological Reasoning, class support, class material. I will put all the slides today. I'm sorry.
I don't have my classes yet, so.
This is very bad. Okay.
It should be uploaded by.
I will upload today, just now, and then I'll make a copy for next time.
I
Okay?
think I'll be able to access my classes by the 22nd. I think that's the deadline for changing for exchange students.
Okay. Okay. But let me know. So if you have any question, don't hesitate. We saw many things.
It's not a question, but did you see the article where they discovered a mutation in CRY2 which made people have a 25-hour
Yeah, that's nice.
clock?
That's nice. That's nice. So this is not FASPs, but it's a DSPS. It's a delayed phase. I think it's the same guy, isn't it? Is it not a guy called Virchow or something?
I'm not sure.
I think it's the same. He has a very big access to human population and to clinical things. It's really, really nice. That's it. So 5 minutes. And then actually there is not only one CK1D, but there are actually two CK1D. CK1D1 and CK1D2, which is the same gene, but two different messenger RNA are made from the same gene, and two different proteins. This is my work here. And then they are completely different. One actually accelerates the clock, the other one slows it down. So it's nice. Anyway, so if you have a question, please ask. If not,